Some key concepts we must know for this lab are wavelength, measured in nanometers, which is the measurement of the photon's wavelength being emitted, emission intensity, which is the associated emission intensity for any given wavelength. This is displayed on the emission spectroscopy on the y-axis. It is proportional to the number of photons of any certain wavelength being emitted. Lambda max, which is the wavelength that has the greatest emission intensity. In other words, the lambda max represents the amount of energy of the photon that is being emitted with the greatest intensity. And the equation in the bottom right relates energy to wavelength. The relationship between the two is inversely proportional, meaning as one increases, the other decreases.
So the research question we're trying to answer is, what determines the emission spectral lambda max? The hypothesis is that the emission spectral lambda max is determined by the observed color of the substance. Let's find out. If this were true, a solution with only one observable color, even if it's composed of two different dyes, would only have one peak on the emission spectra. This is the emission spectroscopy of a solution that looks bright orange to the human eye, but is actually a mix of red and green dye. Instead of only having one peak that should go from 600 to 640 nanometers representing the orange, it has two, so this can't be true.
If we run two other individual emission spectroscopy tests on the green and red dye, we can see that these are the two peaks represented in the first emission spectroscopy of the mixed solution because they're almost identical lambda maxes. The first peak on the emission spectra is green because on this peak, the wavelength with the greatest emission intensity is 504, and on the individual test, it has a lambda max of 508. The second peak is the red dye, as this peak's wavelength with the greatest emission intensity is 614 and has a lambda max of 618 in the individual. This helps us determine that the observed color of a substance does not determine the lambda max on the emission spectra. Rather, it is the molecular composition that determines the lambda max. This is because each substance with its own molecular composition has a unique way in which it moves in between energy levels, and this produces a specific photon of light emitted with the greatest intensity, and therefore determining the lambda max. The molecular composition affects its emission spectra as a substance that has more electrons has more energy levels available for the electrons to be excited to and emit from. The greater number of electrons also allows for more potential higher energy electron configurations as opposed to less electron-dense molecules or atoms. Therefore, the number of available energy levels, differences between them, and the electron configuration determine lambda max.
Of jumping energy levels can be explained with this diagram, where a photon is absorbed by an electron. With the energy from that, the electron is able to be excited to a higher energy state. When the electron regresses to its ground state, or when it relaxes, it emits a photon. This emission is visibly seen as light.
The process of electron excitation and relaxation can also be represented as this type of line diagram. The arrows going upward represent absorption and the ones going downward represent emission. As can be seen, every individual level that the electron goes down emits a specific type of photon. This concept can be compared to an amusement ride called a drop tower, where riders are lifted up a large vertical structure to a certain height and then released, letting them go down in a free fall. The riders can be representative of the electron, and the lifting of them can be compared to the absorption of a photon by the electron. When the electron descends energy levels, it emits a photon. When the riders drop from high to low, the riders emit screams.